Hi, my name is Grace Dharmaraja. I'm a field application scientist at Precision Nanosystems. Before I introduce our speaker, I'd like to thank you all for joining us for this webinar today. I'm happy to see this level of interest in this topic, one that we at Precision Nanosystems are very passionate about. Now, without further delay, it's my pleasure to introduce today's speaker. Our presenter today is Dr. Marc-Andre Dansereau. He will be talking about lipid nanoparticle delivery of siRNA to unravel chronic pain. Marc-Andre completed his undergraduate degree in pharmacology at the University de Sherbrooke, Canada, where he also enrolled for a PhD in the Department of Physiology. In his thesis, he described how a CCR2 antagonist constitutes a potent analgesic in animal models of inflammatory and arthritic pain. Moving later to Trinity College in Dublin for a postdoctoral fellowship, he studied the interaction between peripheral inflammation and dementia. Now Marc-Andre is back at Sherbrooke. He is using RNAi technologies to unravel the dynamic regulation of chemokines in chronic pain. Marc-Andre, it's on to you.
Thank you very much for the introduction, Grace. So today I'm going to talk about how we use lipid nanoparticles to deliver siRNA to better understand how chronic pain established. First of all, we need to realize that pain is a necessary sensation. It allows us to stay away from harmful situations and keep the integrity of our body intact. The primary afferents that detects those painful stimulation all have their cell bodies into the dorsal root ganglia, the DRGs. As you can see in the picture, they form a very diverse population of neurons that are responsive to different kind of stimulation, either noxious or non-noxious, or thermal or mechanical. Following a pain stimulation, the DRGs will transmit this information through the dorsal horn of the spinal cord, where it will then reach the brain. In chronic pain, what is happening is that even if the source of the pain, the injury, is gone, patients still feel the painful stimulation. And there are a lot of reasons for that. There can be neuronal plasticity both at the DRG level and the spinal cord level. There could be glial activation as well that can facilitate the perception of a painful stimuli. There can also be immune cell filtration. So all of these will make it easier to trigger a painful information that our brain will consider as painful. And there are lots of mediators implicated in this chronic pain, as you can see here. All of them are actually studied in different levels to develop new analgesic molecules, but we decided to focus on the CCL2 chemokine, as well as its receptor, the CCR2 receptor. We thought it was an interesting target because it's an important actor of both inflammatory response and nociception. Like all chemokine, CCL2 is a chemotactic cytokine, and it can attract monocytes to the site of injury to promote healing, but also this will trigger an inflammatory response, and this inflammation is enough to facilitate the activation of the nociceptors, the primary afferents that transmit pain through the brain. In addition to that, CCL2 has many effects on the CNS, including pain itself. Using transgenic mice, a group observed that animals who were not expressing the CCR2 receptor developed less tonic pain, less inflammatory pain, and probably most strikingly at all, they did not develop any mechanical hypersensitivity at all following a sciatic nerve injury, which is the main model of neuropathic pain. Accordingly, we previously demonstrated that CCL2 is expressed in the DRGs and is expressed in the same vesicles as substance P, which is a neuropeptide that is being released on painful stimulation. We also showed that intrathecal administration of CCL2 is enough to generate an hypersensitive state. So the animal who receives CCL2 are more sensible to painful stimulation. However, pharmacological approaches with chemokines can be complicated. As you can see here, the chemokine system is a highly redundant system. There are a lot of chemokines and a lot of chemokines receptors. Most of the chemokines can bind to many receptors, and one receptor usually is able to bind multiple chemokines. So when drugs are developed to block these receptors, many of them act on more than one receptor. One of the ways we found to circumvent these issues was to use RNA interference technologies. Small interfering RNA, or siRNA, enter the RISC complex over here, where the passenger strand will be degraded and the guide strand will recognize its target mRNA, leading to its cleavage. Through a collaboration with a company called Integrated DNA Technology, IDT, we were able to use Dicer-substrate siRNA, dsRNA. They entered the siRNA pathway earlier, where an enzyme called Dicer processed the duplex and transferred it afterwards to the RISC. This led to a reduction in off-target effects, an immune response, and is up to 10 times more potent. They designed and tested numerous duplexes targeting different regions of either CCL2 or CCR2, and they test them in vitro to test their efficiency. We used the two that works best, and we used them in our in vivo studies afterwards. However, a lot of transfection regions have some effect when administered in vivo, including the one we used in our in vitro characterization. And this is where the lipid nanoparticles came into effect. The nanoparticle we use were made by Precision Nanosystem on their nano assembler system, and they form around the siRNA. They are uniform in size, and upon being uptaken by cells, they will liberate their content. The siRNA will then be able to find the Dicer complex and reach its target mRNA and knock it down. We validated this theory by injecting nanoparticles intrathecally between the L5 and L6 vertebrae of rats. They contain siRNA against PTEN, a widely expressed phosphatase, as well as a fluorescent dye. We harvested the DRGs of those animals, and we could see, and I will enlarge that in a second, we could see that a lot of neurons could uptake the fluorescent dye, the white dust that you can see. And it was interesting to see as well that different types of cells all seemed to be able to uptake the nanoparticles. So we could see smaller C fibers, intermediate A delta fibers, as well as bigger non-nociceptive A beta fibers. And in blue you can see the nucleus of the glial satellite cells that are all around the neurons. However, we could not observe anything in the spinal cord, and we believe this is due to the distance from the site of injections. So the DRGs at the L5 levels, they project to a spinal cord that is much higher up there, and we believe that the distance from the site of injection prevent any form of uptake from the particle. They just don't reach that far. This was confirmed when we measured the protein expression of P10 by Western blot, where we could see that there was almost no knockdown at all at the L4 and L6 level spinal cord. However, if we took some tissues from the coda equina, which is the region where the spinal cord stopped being the spinal cord, and closer to the site of injection, we could see an almost complete knockdown. The same when we observe it in the DRGs. So this was interesting for us because in the field of chronic pain, DRGs transmit information into the spinal cord, but having tools that selectively targets the DRGs can lead to a better understanding of how those two different organs communicate with each other. The next step was to determine the innocuity of these nanoparticles. In order to do that, we used the formalin test. Briefly, what we do is we inject formaldehyde into the hindpaw of the animals, and we can score the intensity of the nociceptive behavior that this provokes. If the animal don't show any signs of pain, we score it as a 0. If they put slightly less weight on their limb, we score it as a 1. If they lift their limb, we score it as a 2. And if they are actively interacting with their limb, either licking it or biting it, we score it as a 3. The formalin test has 3 different phases. The first one is an early acute phases of moderate pain, followed by an interphase where the animals show almost no signs of pain at all, and followed by the third phase, the inflammatory phase, where the pain behavior lasts for a longer period of time. And basically what we did was we took the area under the curve for this inflammatory period, and we injected intrathecally non-targeting siRNA complex with different transfection regions. And we could observe that a lot of those we used in vitro, most of them being cationic lipid, had an effect on the pain perception of the animals. However, non-targeting siRNA transfected with the lipid nanoparticles, they did not induce any sort of modification in the pain perception compared to animals who receive only intrathecal administration of saline. With this knowledge, we could move on to a model of chronic pain, and we decided to use a model of chronic inflammatory pain, the CFA model. Basically, we inject complete fluid adjuvant into the hindpaw of the animals. This will provoke localized swelling and hypersensitive to pain. In order to measure this hypersensitivity, we used von Frey hairs. They are designed to bend after eliciting a specific pressure, and by using von Frey of different caliber, we can determine which pressure is required to elicit a painful withdrawal reflex from our animals. And this allowed us so you can see here that sham animals, which are healthy animals, will only receive an intraplantar injection of saline. They have a threshold to a mechanical stimulation around 15 grams. When we inject CFA, the threshold of its hindpaw is lower starting on day 1 post-CFA, and it's still lower 7 days after the administration of CFA. What we did afterward was to inject intrathecally a non-targeting siRNA in lipid nanoparticles on day 1 and 2 post-CFA, and we could see that these non-coding siRNA had no effect at all on pain perception of this animal, which was what we expected. When we administered siRNA against CCR2, we were able to reverse the mechanical hypersensitivity up to day 5 post-CFA, but at day 7, these animals had a similar mechanical threshold than animals treated with a non-targeting siRNA. And we had something similar using the siRNA against CCL2, although this effect stopped on day 5. It was interesting to see that CCR2 and CCL2 were not behaving the same. And I'd like to quickly remember that since we are only targeting the DRGs, when we knock down CCR2 is only expressed by the DRG. However, the CCL2 produced by the DRGs will be released toward the periphery or the spinal cord. So in a very simplified way, when we target CCR2, we are mainly targeting the action on the DRGs, while when we target CCL2, we are talking about the action by the DRG. Afterwards, we wanted to see if preemptive administration of the siRNA would completely prevent the apparition of mechanical hypersensitivity in the CFA model. So we injected the siRNA on 24 hours and 2 hours before CFA administration, and we could see that siRNA against CCR2 had a similar effect than when they were injected before CFA, and the same for the siRNA against CCL2. So we still were not able to alleviate the pain on day 7, and this tends to suggest that there is a different pathway completely independent from CCL2 and CCR2 that kind of takes over after 7 days of chronic inflammation. However, if we keep following these animals, we did not inject anything else, we did not treat them with anything else, but if we keep following them on day 14, there we can observe again a huge reduction of the mechanical hypersensitivity induced by the CFA that eventually fades out on day 21. And this suggests a biphasic contribution of the CCL2-CCR2 axis. At this moment, we cannot say if this is due to the earlier knockdown that we induced, and that this knockdown will have a repercussion on those later time point, or if we still have a knockdown due to our 2 injections made on day 1 and day 2. Afterwards, we assessed the action of the siRNA against CCR2 in a different pain paradigm with the lower inflammatory component to see if CCR2 activation was still as important. For this, we used a bone cancer pain model developed in our lab. Briefly, what we do is we inject cancer cell directly into the femur of rats, and you can see at the bottom there that the tumor will grow pretty quickly, and on day 18 it will occupy most of the femoral head, and this would lead to severe bone degradation as well as massive mechanical hypersensitivity as well. And we injected our siRNA against CCR2 on a daily basis, starting on day 11 following the inoculation of the tumor cells, and we could see that we could completely prevent the apparition of mechanical hypersensitivity. This was very interesting for us because even when we used morphine dose that were 10 times higher than was sufficient to alleviate the mechanical hypersensitivity induced by the CFA model, we could not reach such a level of analgesia. So this is really promising and will certainly lead to interesting results in the future. So to conclude, lipid nanoparticles made with the nanoassembler allow the effective transfection of siRNA in vivo. Dicer-substrate siRNA allow for long-lasting and strong behavioral modifications, and these 2 tools combined allow us to identify a very dynamic role of CCL2 and CCR2 in the pathophysiology of chronic pain that we will probably have missed without those tools. Finally, I'd like to thank the people from Integrated DNA Technologies and Precision Nanosystems for providing us with those great tools and solutions to solve our biological questions, as well as all the amazing people in Philippe Sarez's team. And thank you all for your attention.
Okay. So I'll start with one of the questions that was typed into our box there. So I think you kind of addressed this later on. The question might have came a bit earlier. However, they asked, is it possible that your CFA model looks more at acute pain versus chronic pain? Have you examined the effects of siRNA 2 or 3 weeks after CFA injection?
That's a very good question. That is something that we want to do, but at this current stage, the CFA model, at day 21, we start losing a lot of animals because the inflammation is propagating to other hind paw and other limbs. So we need to use a milder model of inflammation and then inject the siRNA at the later time point to see if the CCL2, CCR2 is important once the chronic pain is completely established. Yes.
So just a follow-up from there then, so if you're considering a different model for chronic pain in terms of when you'll be making the injection, will you also be looking at whether other receptors are involved? So you're focusing right now on CCR2 and CCL2 and its ligand. Are you interested in other such receptors that you showed earlier on in your introductory slides?
Well, it would be interesting to test all of them, really. There's each time you're interested in how inflammatory markers or cytokines contribute to chronic pain, all of them seems to be solely responsible for chronic pain, which is not the case actually in a behavioral model. So it's hard to take into account all those interactions between all of them. What we could do, however, is to maybe to study receptors that alleviate pain and try to overexpress them. That's something that we'd like to try as well.
Okay, perfect. So thank you so much for your time, Marc-Andre. So I think we'll be wrapping that up with your questions, and we'll answer any questions that might have come through via our question box through an email. So thanks everyone who joined us today. If we didn't get to your question, as I mentioned, we'll follow up with an email after the event. Again, if you have any questions about the nanoassembler or would like to learn more about it, please visit us at www.precisionnanosystems.com, and don't forget to follow us on LinkedIn and Twitter. So from myself, Grace, as well as Marc-Andre, have a wonderful rest of your day, and thank you so much for your time. Cheers.